Hello students, in this module let us learn about Introduction to Lab Safety, Micropipette use, solutions, dilutions and unit conversions. Let us first see the introduction. This lab module introduces the basic concepts of laboratory safety, micropipette use, solutions and dilutions and unit conversions. The part on lab safety discusses the appropriate actions suitable for students upon entering or exiting the lab. The section on use of micropipettes introduces the basic skills for handling micropipettes and the precautions to be taken while using micropipettes. Further, the module discusses the methods for preparing different types of solutions and also the laboratory security and safety procedures to make serial dilutions from the stock solutions. Finally, the module winds up with the study of conversion factors and the correct use of units. First, let us take up laboratory safety. Cell biology laboratories commonly have hazardous supplies and equipments which require proper knowledge of handling to avoid hazards. Anyone working in the lab must know the safety rules and should pay attention to their work. The key components to lab safety precautions and procedures that commonly apply in a cell biology laboratory may be discussed under the following sections. The first one is laboratory security. Since a typical cell biology lab may be using some hazardous materials, it is important to prevent unauthorized entry to the labs, secure the hazardous materials against theft, as well as ensure compliance with security regulations to prevent unwanted accidents caused by exposure to the highly hazardous materials. Next is proper handling and storage of chemicals and reagents. This requires certain safety measures which are to be undertaken while handling and storage of chemicals and reagents used in the lab. One such measure is labeling of the chemicals and reagents. Labeling should be done in such a way that anyone can identify the chemicals and reagents by seeing the labels. Moreover, the lab should have a file of material safety data sheet on every chemical which describes the specific properties of a chemical and may include the identity of the chemical, hazardous chemical or ingredient, physical chemical characteristics of the chemical or reagent, data on fire and explosion hazard, data on reactivity, health hazards specifying toxicology data, and the precautionary measures for safe handling and use of the chemical. Next is the disposal of hazardous chemicals and biological materials. The disposal of hazardous chemicals should be done according to the rules and regulations of the state or country. The disposal of hazardous chemical and biological waste should be discussed at the beginning of every lab. For disposal of biological hazards, the biological organisms are commonly contained in sealed autoclavable bags and autoclave at high temperatures and pressure to kill the organisms completely. While in case of hazardous chemicals, they are usually disposed in disposal containers kept in the fume hood. The next is micropipette use. Micropipettes are used to measure and transfer small volumes of solution or liquid. These small instruments have a screw for adjusting the volume of air space in the barrel and works by screwing the plunger in or out of the piston. They also have a digital display readout that displays the corresponding volume. When the plunger is depressed, it displaces the specified volume of air from the piston, and when the micropipette with a tip and depressed plunger is dipped into a fluid and the plunger is released, the plunger creates a vacuum and an equal volume of fluid is drawn into the tip. This withdrawn fluid in the tip can then be expelled by depressing the plunger again. Micropipette may come with a set fixed volume or maybe with a volume range, such as the P10 with a range of 0.5 to 10 microliter, P20 with a volume range of 2 to 20 microliter, P200 with a volume range of 20 to 200 microliter, or a P1000 with a volume range of 100 to 1000 microliters. The general steps for using a micropipette are as follows. First, adjust the desired volume by rotating the screw. Now, place the end of the micropipette on a proper size tip and set it firmly. Always hold the micropipette in the palm by wrapping the fingers around the barrel and regulate the piston with the thumb. The micropipette should always be held vertically when filling it. Most micropipettes commonly have a two-position plunger with friction stops, which can be felt when pressed with the thumb. When the plunger is depressed to the first stop, it measures the desired volume, but when the plunger is depressed to the second stop, it blows out an additional volume of air to expel any remaining solution in the tip. For withdrawing a fluid, the plunger should be depressed to the first stop, the tip is dipped into the solution, and the plunger is gradually released to draw the fluid into the tip without any air space at the end of the tip. The excess droplets adhering on the outside of the tip can be get rid of by sliding out the micropipette tip along the inner wall of the reagent tube. For expelling the fluid into another tube, gently touch the tip of the pipette to the inside of the tube by holding the tube between the thumb and the forefinger at eye level to observe the change in the volume of fluid level in the pipette tip, and depress the plunger to the first stop slowly to expel the sample, and then depress the plunger to the second stop to blow out the remaining bit of fluid. Eject the tip or manually remove it, and always use a fresh tip for each new reagent to be pipetted. The following precautions should be taken when using a micropipette for accurate measurement and maintenance of the micropipettes. First, the volume adjuster screw should never be rotated beyond the upper or lower range of the pipette. Always avoid inverting the micropipette or laying it down with a filled tip to prevent fluid from running back into the piston. After withdrawing or expelling the fluid, never let the plunger snap back as it can damage the piston. The barrel of the micropipette should never be immersed in fluid. Let us now take up solutions. Solutions may have different units of concentration or the amount of solute in a solution. Solutions containing high amounts of solutes are usually prepared by initially dissolving the solutes in a small volume and then making up the total volume of the solution to the designated volume. Some commonly used concentration units for solutions are as follows. The first one is weight per volume solutions. A weight per volume solution has a certain amount of a solute in a certain volume. For example, milligram per ml or gram per ml or gram per liter. Next are the molar solutions. A molar solution contains a specific number of moles of the solute in 1000 ml or 1 liter of the solvent. A solution containing 1 mole of a solute dissolved in 1000 ml is said to have a concentration of 1 molar. Solutions containing smaller concentrations of solutes may be expressed as millimolar or micromolar solutions. Next are the percent solutions. In a percent solution, the concentration of a solute is expressed as a percentage of a total of 100 parts. For liquids, the percent concentration are usually expressed in terms of a volume per volume ratio. For example, 1 ml of solute in 100 ml of solvent has a concentration of 1% solution. On the other hand, for solid solutes, percent concentrations are usually given as weight per volume ratio. For example, a 2 gram solute dissolved in 100 ml of solvent has a 2% solution concentration. Next, let us take up dilutions. Dilution factors are conveniently used for using a concentrated stock solution in a dilute form in an experiment by diluting with a liquid called the diluent. The amount of dilution or the dilution factor is usually expressed in terms of the total number of unit volume in which the solution is to be diluted. For example, a solution with 5 dilution factor has 1:5 dilution, that is, dilution achieved by combining 1 unit volume of the solute or concentrated stock solution with 4 unit volumes of the diluent. Hence, dilution factor will be equal to 1 plus 4, that is 5. A serial dilution is a series of simple dilutions which begins with small initial volume of stock solution and amplifies the dilution factor by diluting the solution from the previous step. The total dilution factor in a serial dilution is given by the product of the individual dilution factors in the steps of the series of dilution. That is, final dilution factor will be equal to dilution factor 1 multiplied by dilution factor 2 multiplied by dilution factor 3 and so on. For example, if an initial dilution step combines 1 ml of a stock solution with 2 ml of diluent, it makes a 1 out of 3 dilution. Further, if 1 ml of the diluted solution is mixed with 2 ml of diluent, it makes a 1/3 dilution of a 1/3 dilution or a 1 out of 9 dilution, and when 1 ml of this diluent solution is again mixed with 2 ml of diluents, it makes a 1/3 dilution of a 1/9 dilution or a 1 out of 27 dilution. Next is the conversions. Conversions of units often become necessary in calculating different units of factors and may involve the conversion between different metric units or between metric and English or American units. Accurate conversion of units may be calculated by using conversion factors or dimensional analysis. A conversion factor is a simple ratio that expresses the relationship between two units. For example, 2.54 centimeter can be written as 1 inch since there are 2.54 centimeter in an inch. Similarly, 0.0 0.0000837 meters can be converted to 8.37 micrometers. Let us now see the conclusion. The lab safety precautions and procedures commonly followed in a typical cell biology laboratory encompasses safety measures related to laboratory security, handling and storage of hazardous chemicals and reagents, and also proper disposal of hazardous chemicals and biological materials. Micropipettes are small equipments commonly used in laboratories to measure and transfer small volumes of solution or liquid, and knowledge about their use and precautionary measures are essential to work in a cell biology lab. The different concentration units used for solutions such as weight per volume solutions, molar solutions and percent solutions are commonly used. Concentrations and preparations of solutions and dilutions or serial dilution of solutions are also commonly involved in biology experiments. Last but not the least, a brief idea on conversion of units will help in interconversion between different units or unit systems, which will help in calculating different units of factors. Thank you.